Aim or objective: to investigate biogenic preparation of zinc oxide, copper oxide, and selenium nanoparticles using the marine brown alga Sargassum schwartzi. The objectives of the present study are fourfold. Firstly, biogenic preparation and physico-chemical characterization of zinc oxide, copper oxide, and selenium nanoparticles using marine alga Sargassum schwartzi. Secondly, to isolate and molecular characterization of Vibrio parahemolyticus using 16S RNA gene sequence and PCR analysis. Third, to study antibacterial activity against newly isolated bacterial pathogen Vibrio parahemolyticus using biogenic prepared zinc oxide, copper oxide, and selenium nanoparticles. Fourth, interaction of nanomaterials, zinc oxide, copper oxide, and selenium with Vibrio parahemolyticus has been carried out using SEM EDAX studies. Introduction: Nanomaterials, materials processing at minimum one external dimension measuring 1 to 100 nanometer, particle size of at least half of the particles in the number size distribution must measure 100 nanometers or below. Nanoparticles: a particle of matter that is between 1 and 100 nanometers in diameter. Nanotechnology, a field of research and innovation concerned with building things generally, materials and devices on the scale of atoms and molecules. Aquaculture is one of the largest food-producing sectors across the globe. Disease is considered to be one of the major limiting factors in aquaculture production worldwide. Aquaculture industry, particularly shrimp culture, has been continuously suffered by various diseases that are caused by bacteria and viruses, leading to high mortality rate and huge economic loss. Bacteria cause for high mortality rate in shrimp industry worldwide. Early mortality syndrome in penile shrimps is named as acute hepatopancreatic necrosis disease infected by Vibrio parahemolyticus has been reported and caused highest mortality, which is 100% within the 30 days of culture. Materials such as plant extracts, feeds, probiotics, immunostimulants, recombinant immune-related proteins, silver nanoparticles have been used for the treatment of AHP&D infection. Metal oxides used for different applications due to the remarkable properties. Zinc oxide, which is 2,6 semiconductor with distinctive properties, have applications such as sensors, biosensors, photocatalysts, solar cells, and antibacterial agent. Copper oxide have potential applications such as biocidal activity, catalysis, and antibacterial activity. Selenium is a well-known structural component of the active center of various antioxidant enzymes and functional proteins. It can contribute in different redox reactions due to its multiple oxidation states. Further, it is an essential trace element for human due to its pro-oxidative and antioxidative effects. Although various chemical and physical protocols are used for the preparation of metal nanoparticles, various green and affordable preparation techniques have been expanded using plants, microbes, and their therapeutic applications such as antimicrobial activity, anti-cancer, anti-diabetic, cardiovascular, agriculture applications. Among the natural resources, seaweeds have potent resources use of natural, cost-effective, high stability, and biocompatible materials as stabilizing agents. in the preparation of metal nanoparticles. Further, these bioagents are accomplished of providing enough stability in various therapeutic applications even in the presence of electrolytes and under conditions of different pH. Nanoscience and technology has a remarkable potential to transform agriculture and allied sector, which includes aquaculture and fisheries. It can provide new tools and techniques for rapid disease diagnosis and to improve the ability of culti cultivable species to uptake nutrients, hormones delivery, and vaccine delivery. This picture is the graphic design of the whole process, how the material is prepared and affected on the Vibrio parahemolyticus. First of all, Vibrio parahemolyticus is taken. After that, isolation of Vibrio parahemolyticus, PCR confirmation, Vibrio parahemolyticus cells, seaweed, preparation of nanoparticles, characterization of nanoparticles obtained, bacterial activity, nanoparticles, and bacterial death. Materials and methods: seaweed collection and preparation of extract. Brown macroalga S. swartzi was collected from Mandapam rameswaram from India, washed with distilled water to remove the debris and other contaminants, dried in shade and powdered using an electronic blender. About 10 grams of powdered seaweed sample was dissolved in 1 liter of distilled water and incubated in a shaker overnight under room temperature. Extracts were filtered using Wattsman filter paper number 1, and the filtrate was used for the preparation of nanomaterials. Preparation of zinc oxide nanoparticles. Seaweed stabilized zinc oxide nanoparticles was synthesized using co-precipitation method. Macroalga extract 80 ml was added with 400 ml of 0.1 molar concentration of zinc acetate and mixed well using a magnetic stirrer at 70 degrees centigrade for 20 minutes. About 400 milliliter of 0.2 molar NaOH aqueous solution 400 milliliter was added drop-wise into the reaction solution under continuous stirring condition for 3 hours. After 3 hours, white precipitates were obtained, thus confirming the formation of zinc oxide. Twice the precipitates were collected, dried, and allowed for the calcination at 400 degrees centigrade for 4 hours to obtain the purest form of zinc oxide nanoparticles. Preparation of copper oxide nanoparticles. Copper oxide nanoparticles were prepared at room temperature through co-precipitation technique. In this experiment, 0.1 molar aqueous solution of copper acetate along with 40 ml of algal extract was kept under constant stirring for 30 minutes. Then 0.2 molar of NaOH aqueous solution was added drop-wise into the reaction mixture and allowed to proceed for 4 hours after complete addition of NaOH. Further, the reaction solution was incubated overnight at room temperature for settlement of nanoparticles as precipitates. The precipitates were separated using centrifugation at 5000 × G for 10 minutes, washed several times with deionized water and dried at 80 degrees centigrade for 12 hours. The dried powder was calcinated at 400 degrees centigrade for 4 hours to obtain the copper oxide nanoparticles. Preparation of selenium nanoparticles. For the preparation of selenium nanoparticles, selenious acid was mixed with 100 milliliter of algal extract under magnetic stirring condition, then 150 milliliter of freshly prepared ascorbic acid 1.056 gram solution was slowly added into the reaction mixture until brick red precipitates were formed. Further, the solution was centrifuged at 5000 G for 20 minutes and the pellet was washed with deionized water and dried to obtain selenium nanoparticles. Molecular identification of Vibrio parahemolyticus. The isolates were cultured in tripartite soy broth (TSP) media overnight, and the genomic DNA was extracted by phenol chloroform extraction technique. The genomic DNA was quantified using a biospectrometer and stored at 80 degrees centigrade for further analysis. 16S ribosomal RNA partial sequence was performed and the sequence homology of extracted 16S RNA 16S ribosomal RNA gene sequence was analyzed by NCBI BLAST. The similarity matrix of 16S ribosomal RNA gene sequence of strain was obtained using phylogeny interface package and the sequences were aligned using the clusteral X program. The phylogenetic tree was constructed based on neighbor joining method using the Kimura 2 parameter model by the Mega 7 software package and tested by bootstrap analysis based on 1000 resamplings. The evaluationary distances were computed using the maximum composite likelihood method. Newly identified bacterial sequence was submitted to the NCBI to obtain accession. Antibacterial activity using in vitro test. Diffusion was performed on Mueller-Hinton agar plates with 2% sodium chloride spread with 100 microliter Vibrio parahemolyticus bacterial suspension. Under aseptic conditions, well 5 mm was made and filled 10 microliter with different concentration of ultrasonicated biogenic nanomaterials and control was maintained in the absence of nanoparticles. The plates were incubated for 24 hours at 28 degrees centigrade. The inhibition zone was measured with zone of reader. The effect of nanoparticles on the growth rate of Vibrio parahemolyticus grown in Mueller-Hinton broth was studied. The bacterial suspension was treated with different concentration of zinc oxide, copper oxide and selenium nanoparticles and incubated at 28 degrees centigrade plus minus 1 degree centigrade. The growth rates were determined by measuring the optical density at 1600 nanomaterials 1600 nanometer using ultraviolet spectrophotometer and each measurement was made at an interval of 3 hours. SEM and EDAX studies the interaction and morphological change in Vibrio parahemolyticus cells on treatment with and without zinc oxide, copper oxide, and selenium nanoparticles were carried out by SEM with EDAX studies with some modification. The elemental composition and elemental mappings were carried out using energy dispersive X-ray analysis. XRD analysis. Picture A shows. This picture shows the XRD analysis of zinc oxide nanoparticles. Picture B shows copper oxide nanoparticles X-ray XRD analysis. Picture C shows selenium nanoparticles XRD analysis. SEM and EDAX analysis. Figure 3 AB SEM image and EDAX spectrum of zinc oxide nanoparticles. Figure 4A B SEM image C EDAX spectrum of copper oxide nanoparticles. Figure 5A B SEM image EDAX spectrum of selenium nanoparticles. Conclusion: the zinc oxide, copper oxide and selenium nanoparticles have absorption at 395 nanometer, 260 nanometer and 233 to 258 nanometer in DRS ultraviolet spectrum. X-ray diffraction spectrum confirms the 2 theta values corresponding to the presence of zinc oxide, copper oxide and selenium with JCPDS SEM micrographs of zinc oxide, copper oxide and selenium nanoparticles which shows spherical morphology. The new bacterial strain were isolated from infected shrimp sample and identified as Vibrio parahemolyticus based on 16S ribosomal RNA sequencing and presence of specific virulent genes, which are TOX, R and TLH. The biogenic nanomaterials exhibited potent antibacterial efficiency against Vibrio parahemolyticus. The interaction of nanomaterials with Vibrio parahemolyticus was analyzed using SEM, which revealed that the attachment of nanoparticles on the surface of cell membrane causes non-viability of cells. The present study confirms the enhanced antibacterial efficiency of seaweed stabilized nanoparticles, which are zinc oxide, copper oxide, and selenium. Hence, the prepared nanoparticles can be formulated as a bactericidal material against the aquaculture pathogens in efficient disease control. Figure 6. This picture shows the systematic diagram showing the nanomaterials interaction and their mechanism of antibacterial activity. These are the cells of Vibrio parahemolyticus, which are interacted with zinc oxide, copper oxide, selenium nanoparticles. This is a cell which is interacted with nanoparticles. Literature. This is the literature from which we have followed this data. Thank you.